The cell is the basic unit of life, a tiny structure that performs every activity needed to keep us alive. When we observe a cell under the microscope, the first thing we see is its outermost layer, the cell membrane. Inside this membrane lie several organelles, and interestingly, most of them are surrounded by membranes too, made of similar material. This cell membrane acts like a bodyguard, protecting the cell and controlling what enters or leaves. It performs many vital functions, so today, let's take a closer look and understand the composition and structure of this remarkable barrier. In 1972, scientists S. Jonathan Singer and Garth Nicolson proposed the Fluid Mosaic Model of the cell membrane. According to this model, the membrane is made up of a double layer of phospholipids with proteins embedded within it, and it is fluid rather than solid. The phospholipid bilayer forms a fluid, C, in which specific proteins float, much like icebergs drifting in water. Because it's fluid, the membrane is in a constant state of motion, shifting and changing while still maintaining its overall structure. The term, mosaic, refers to the variety of different proteins scattered throughout the phospholipid layer, giving the membrane a patchwork appearance. The following are the main components found in a typical cell membrane. Phospholipids form the basic framework of the cell membrane, a bilayer where the molecules are arranged tail to tail. Each phospholipid molecule has two distinct parts. The hydrophilic head, which means water-loving, faces outward toward the extracellular material and inward toward the cytoplasm, since both contain water. The hydrophobic tails, meaning water-fearing, point inward, facing each other and away from the watery surroundings. This unique arrangement creates a selective barrier, allowing lipid-soluble molecules to pass through easily while blocking most water-soluble substances. These purple-colored structures are proteins. In the cell membrane, different types of proteins are embedded within the phospholipid bilayer, and each performs a unique function. Let's explore the main types of membrane proteins and understand how they help the cell carry out its vital activities. Proteins in the cell membrane are divided into three main types, based on how they are attached or positioned within the membrane. These are integral proteins, peripheral proteins, and lipid-anchored proteins. Integral proteins, also known as intrinsic or transmembrane proteins, are one of the major types of membrane proteins found in the cell membrane. These proteins are embedded deep within the phospholipid bilayer, and many of them span completely across the membrane, from the outer surface to the inner side of the cell. Because of this arrangement, they interact with both the hydrophilic heads and the hydrophobic tails of the phospholipids. Their unique positioning allows them to perform some of the most vital functions of the cell membrane. They help in transporting materials, receiving signals, and maintaining communication between the inside and outside of the cell. Now, based on their functions, integral proteins can be divided into several categories. First, channel proteins. They form open pores that allow specific ions or molecules to pass freely across the membrane, maintaining balance inside the cell. For example, ion channels like sodium, potassium, or calcium channels help regulate the movement of these charged particles, which is essential for nerve impulse transmission and muscle contraction. Second, carrier or transport proteins help move larger or charged molecules that cannot diffuse directly through the lipid layer. Depending on how they transport materials, they work in three different ways. Uniport moves a single type of molecule in one direction. For example, a glucose uniporter helps carry glucose into the cell. Symport moves two different molecules in the same direction. For instance, the sodium-glucose symporter transports both sodium ions and glucose together into the cell. Antiport moves two different molecules in opposite directions. A common example is the sodium-potassium pump, which pushes sodium ions out of the cell while bringing potassium ions in. Receptor proteins act like the cell's communication antennas. They receive chemical signals, such as hormones or neurotransmitters, from outside the cell and trigger specific responses inside. For example, insulin receptors on the surface of a cell detect the hormone insulin and signal the cell to absorb glucose from the bloodstream. In this way, receptor proteins help the cell respond and adapt to changes in its external environment. Enzymatic proteins act as biological catalysts on the cell membrane. They speed up important biochemical reactions that take place on the cell's surface. For example, ATP synthase in the inner mitochondrial membrane helps produce ATP, the main energy currency of the cell. These proteins ensure that vital cellular reactions happen quickly and efficiently, keeping the cell active and functional. Peripheral proteins are not embedded within the phospholipid bilayer. Instead, they are loosely attached to the inner or outer surface of the membrane. They connect either to integral proteins or to the polar heads of phospholipids through weak interactions. Because of this, they can be easily removed without damaging the membrane structure. Functionally, peripheral proteins play supporting roles. They help stabilize the membrane, maintain the cell's shape, and assist in cell signaling and enzymatic activities. For example, some peripheral proteins anchor the membrane to the cytoskeleton, giving structural support, while others participate in signal transduction by transferring messages inside the cell. Lipid-anchored proteins are special types of membrane proteins that are covalently attached to lipid molecules within the cell membrane. Unlike integral proteins, they do not span the membrane. Instead, they sit on the surface of the lipid bilayer and stay connected through their lipid anchor, which keeps them firmly attached to the membrane. These proteins play important roles in cell signaling, recognition, and anchoring enzymes near the membrane surface where reactions occur. These small yellow dots are cholesterol molecules. They fit between the phospholipid tails and play several important roles, keeping the membrane fluid yet stable, preventing it from becoming too rigid in cold conditions, and reducing permeability so that only specific substances can pass through. Cholesterol also supports the proper functioning of membrane proteins, maintaining the perfect balance of strength and flexibility in the cell membrane. These antenna-like structures you see on the surface are carbohydrates. They attach to either proteins or lipids, forming two important structures. When attached to proteins, they form glycoproteins, and when attached to lipids, they form glycolipids. Together, these carbohydrate structures play a key role in cell recognition and communication. So that's all for today's video on the composition of the cell membrane. I hope it was helpful and that you learned something new today. If you did, don't forget to like, share, and subscribe to the channel for more biology lessons. Thanks for watching, and I'll see you in the next video.